Hello, everyone. My name is Ashley Fernandez, and I'm a publishing professional at Inago and have been actively involved in developing comprehensive and easy to understand resources for academic researchers and assisting them in their publication journey. On behalf of Inago Academy, I welcome you all to Inago's Cancer Research webinar in collaboration with Anti-Cancer Inc., the world's leading preclinical cancer contract research organization. This session will focus on insightful discussions on the mechanism of methionine addiction, popularly known as the Hoffman effect. Furthermore, we will also understand how this fundamental hallmark of cancer can be targeted therapeutically. I would request all attendees to kindly send us their queries or questions at any point of time using the Q&A tab in your Zoom control panel, and our speaker would be happy to answer all your questions at the end of this webinar. Today, we have with us Dr. Robert Hoffman, President, Chairman of Board, and CEO at Anti-Cancer Inc., with 55 plus years of research in cancer. Dr. Hoffman is a veteran scientist, an accomplished and globally acclaimed researcher with prolific research achievements. To date, Dr. Hoffman has published over 1,100 research papers with nearly 52,283 citations to his credit. Currently, he is appointed as the professor in the Department of Surgery at the University of California, San Diego. Furthermore, he is also the editorial board member of Clinical Cancer Research, Journal of Fluorescence Guided Surgery, In Vitro Cellular and Developmental Biology, and Anti-Cancer Research. Dr. Hoffman, I formally welcome you to this session. I hope you're having a great day.
Thank you, Ashley. I'm very happy to be able to be here, and I welcome all the audience. Thank you for taking your time to listen to this webinar. The people at Inago did a great job to organize it. I didn't do anything. They're really great.
Thank you very much.
We have an audience, and I just got a little message on the screen from my classmate, Steve Clark, who's a pioneer in this field. And Steve, I'm so happy to hear from you and that you've joined us. Your work will always be remembered. Thank you. So I want to talk a little bit about, but first I'm going to just mention what's methionine addiction. So this is a characteristic, we think, maybe of every cancer. We have to demonstrate that further and further, but so far we found no exceptions. And the methionine addiction is found in the cancer that they require exogenous methionine in order to grow, for example, in vitro, even though they're synthesizing a lot of methionine. They can, so this was this, I don't know if you can see this, the methionine addiction was discovered by us in 1976. We published anyway, it's in PNAS volume 73, page 1523. So here's a very, very old paper. And so you may say, okay, methionine addiction, you claim it's a fundamental hallmark of cancer. You claim it's so important. How come most of us never heard of it? Well, even though we've done a lot of research, we and others over the years on methionine addiction, until about a year and a half ago, it remained obscure. There were papers that came out every 10 years or so about the hallmarks of cancer. There was one in 2000, and it's, I think, published in Cell, one in 2001, one in 2011, the hallmarks of cancer. Of course, methionine addiction was not even mentioned, and so many people don't know. So why is that? And I think I have some explanation, and I would like to share that with you because I think it gives some history into cancer research. Every field has a history, and without that history, the field wouldn't be what it is. If it weren't for Copernicus, there would be no astronomy. So the history is very important, and it explains a lot about until recently, methionine addiction has been obscure. So I want to give you a little bit of background. I want to show you a picture of a man. This man, his name was Otto Warburg, W-A-R-B-U-G, Otto Warburg. He did research in Germany during the Nazi period. And the Warburg effect is named after him. And the Warburg effect was something very unusual and counterproductive, but it seemingly is also a very frequent, if not universal, characteristic or hallmark of cancer, in that cancer cells use abnormally high amounts of glucose. They use the glycolysis pathway even in the presence of oxygen. In Warburg's time, he thought that was maybe because of that the mitochondria in the cancer cells were defective, and that's probably not true. But basically, the main features of the Warburg effect that he published on, he got a little bit older as he went on, he became to look like that. And his classic paper in English is published about 30 years after his original paper in German, and it's called On the Origin of Cancer Cells. And it's a very interesting paper. It's Science on the 24th of February, 1956. So for most of you, that seems like 1,000 years ago, not for me. And so he explained what his effect was, the Warburg effect, the cancer cells using this large excess of glucose. And this is one of the bases of PET imaging. They use a radioactive analog of glucose, and it lights up cancers because cancers are so hungry for glucose. And he was very famous for this. He won the Nobel Prize in 1931. And his history, his personal history, is very interesting. He survived Nazi Germany, even though he was Jewish, because Hitler protected him. And I think maybe Hitler was so afraid to get cancer, but Warburg was protected, and he became the most famous cancer researcher probably in history, and I think deservedly so. So in the 30s and 40s and 50s, maybe even into the 60s, his Warburg's theories were the paradigm. This was the dominant study in cancer, like glycolysis and what's altered in glycolysis in cancer. This was many people studied this. The leading researchers studied this. You were in the in-group if you studied this. So then things started to change in the 1960s, and this is very important. And you'll be surprised, maybe, to know one of the basic reasons there was a change in the 1960s. And you're really going to be surprised what I say, because in the 1960s, the American war against Vietnam became very escalated. The government was drafting people, including people my age. I had a draft card. And so many people were afraid of being drafted and sent to Vietnam and got killed. So the government made a special exception for physicians. And they said, okay, if you go to NIH, the National Institutes of Health, you don't have to, we won't draft you. And at that time, at the National Institutes of Health, I have to share with you this name. His name was Robert Huebner, Huebner. Okay, so what's the significance of Robert Huebner? Huebner was a great virologist. So all of the young doctors who came to the NIH, and they were very, very bright. Some of them very famous to this day: Steve Rosenberg, Stu Aronson, many, many, many. Some still stayed at NIH. And at that time, they were under the tutelage of Dr. Huebner. They all became virologists. So there's only so many things you can study in virology. You can study this disease, polio. Now, of course, we study COVID. But these doctors, I think many of them were interested in cancer. So around 1970 or so, we had a president in America by the name of Mr. Nixon. And Nixon was very much trapped in this Vietnam War. He decided to make another war. He called it the War Against Cancer. And he gave, it was through his great efforts that he allocated many, many billions of dollars to the NIH, in particular, the National Cancer Institute, to study the virology of cancer. It was called the Viral Carcinogenesis Program. And for the first time in the United States, there was very big money in cancer research. And the dominant paradigm through Dr. Huebner, and he was a great big boss, and through the large sums of money that Nixon's efforts led to being funded at the National Cancer Institute, the new paradigm that basically succeeded the great paradigm of Warburg was that viruses caused cancer. The main cause of cancer was virus infection. So all the big names and not so big names started to study viruses and cancer. At that time, I was still a student in Harvard in the biology department. And my favorite teacher was Jim Watson. And Dr. Watson was a great advocate of studying viruses and cancer. And I thought that was the right way. And around that time, there was another great discovery in viruses. It was by a man called Peter Duesberg. And Dr. Duesberg, wrote a paper in 1970, PNAS, volume 67, page 1673. You can get a little look here. Duesberg is D-U-E-S-B-E-R-G. And this was the discovery of the first oncogene. And this gene was called SARC or SRC, and it was the gene of the Rous sarcoma virus. And this paper describes the isolation of the RNA for this gene. It was a momentous discovery. And many years later, 45 years later or so, was the 100th anniversary of the Proceedings of the National Academy of Sciences. And they designated 100 great papers over 100 years. And Duesberg's paper was one designated, and it was summarized in 2015 by a scientist called Klaus Bister. And you can read that in PNAS, volume 112, December 15th, 2015. So it was a great paper. So this kind of just settled everything. Okay, viruses cause cancer. Viruses have an oncogene. It made sense. It made sense. This was cancer. The cause of cancer was solved. Everybody thought so. So Duesberg discovered the first oncogene. It was in the Rous sarcoma virus. It was assumed that there were many, many other kinds of viruses that also could cause cancer. The Rous sarcoma virus caused cancer in chickens. Rous himself, I think, won the Nobel Prize. So it was a great discovery. Cancer was on the way to being solved, on the way to being cured. everything going really good. And I'll just tell you a little story because science never goes in a straight line. And this is a picture of Dr. Duisberg here. And this picture is in Nature, volume 380, March 1996. And it's even though Nature is a British journal, they used French in the title. Adieu Duisberg, goodbye Duisberg. Why? Because Duisberg, after he discovered the oncogene in the Rausch-Sargoma virus, many years later said this is not the cause of cancer. And that was heresy. That was a bad thing to do. And later, he questioned whether HIV caused AIDS or not. That was a very bad thing to do. And so Duisberg made many, many enemies, and he has somewhat of a similar history to Giordano Bruno. Read about him. He was burned at the stake in the middle of Rome in the year 1600 because he believed the other stars in the sky were similar to the sun. But he questioned the dominant paradigm. Quite a dangerous thing. Duisberg was not physically burned, but his character was burned. And things haven't changed so much over 400 years. Okay, so here's we are. Duisberg discovered the oncogene in the Rausch-Sargoma virus. The whole United States cancer research establishment was looking for viruses in every patient's cancer that they could, but they had trouble. They had trouble finding viruses in many of the cancers, maybe most of the cancers. How could it be? And so then some questions started to come up. Why we couldn't find viruses. People, I found a little bit of viral RNA, little pieces and snippets, but it was tough. They couldn't. So people were in a quandary. They thought the whole cancer problem was solved, but it wasn't. Cancer's not so simple. It's not simple now. It's not simple then. Never was simple. Then another amazing thing happened. In 1978, a scientist you never heard of called Dimitrios Spandidos, S-P-A-N-D-I-D-O-S, Dimitrios Spandidos, published a paper in Cell, 1978, volume 12, page 675. And he published another paper in Nature. Here, this is Spandidos. He's a Greek working in Canada in the lab of Lou Siminovich. And this is Nature, volume 271, 1978. And what Spandidos found was he could transform Chinese hamster ovary cells with chemicals so that they became more malignant. He could take the DNA from the Chinese hamster ovary cells, put the DNA into more normal-like cells, and they became malignant. Now, these Chinese hamster cells from which Spandidos isolated this DNA, they didn't have any viruses. This was all cellular DNA that was causing cancer in another organ, another cell line, and this became a mystery. Now, I have to just go back a few years to 1973. Only a few years before that, the first transfection experiments were published, mainly by McBride and Ozer, O-Z-E-R. And I'm sorry, I don't recall the journal right offhand. But so transfection, which was an old story in bacteria, now became known to be possible even in mammalian cells. DNA could be taken from one cell and transformed to another. In the beginning, some genes were transferred, some were linked genes, but Spandidos found, wow, you can transfer what are later been named oncogenes. So people start, wait a minute, cells have their own oncogenes. They don't need a virus. And Spandidos became a very hot item. everybody wanted to hire Sponditos. So he was working in Toronto, and this was around 1978, and he was invited to the Boston bio community, which is very active, and I was still around there. And he gave a number of seminars around the Boston area, and he gave one seminar at MIT, and I went. I was in Harvard, MIT was just down Massachusetts Avenue, a couple of miles. Okay, I went. And sitting next to me was somebody you've heard of, Bob Weinberg, Robert A. Weinberg, Bob Weinberg. And we were both listening. We were sitting together. We're listening to Sponditos. Everybody wanted to hire Sponditos. And Weinberg was not happy. He said, why they recruit him when I'm up for tenure? Why they need him? They can have me, he said. I said, yeah, okay. So it was left at that. Sponditos talked about how he could transfer DNA from one cell type to another. And if the donor was malignant, then the recipient became malignant. Oh boy. A few months later, all this rumors started going around, at least Harvard and MIT. Hey, did you hear? Sponditos made up all the data. It's all fake. He made it up. It was just so shocking because he was maybe the hottest scientist in North America. I don't know, maybe the world. He made it up. He's been kicked out of the lab. His big boss, Dr. Siminovich, kicked him out of the University of Toronto. He had to go back to Greece. What? How can that be? And it was true. He got accused of making up the data. And let me just tell you as a side, he has about 3 Cell papers, Nature paper, all in the 70s. You've never heard of those papers, but if you go on Google Scholar, you'll find them. And I'll tell you another thing. They were never retracted, never. And there was never an investigation into if Sponditos really made up the data or didn't make up the data. It was never proven one way or another. And those papers still stand, but nobody cites them. It's like a taboo. You're not allowed to cite Sponditos papers. It's like an unwritten rule. Okay, then the story continues. And we're starting to enter the modern world. And in 1979, 1 year after Sponditos' paper, 1 year after Bob Weinberg sat in the seminar listening to Sponditos next to me, a paper comes out in PNAS from Bob Weinberg's lab, PNAS volume 76, page 5714. And this paper says, passage of phenotypes of chemically transformed cells via transfection of DNA and chromatin. Exactly what Sponditos showed 2 and 3 years before, but instead of CHO cells, which are famous, this paper used another cell line, NIH3T3 cells, kind of similar. One's from a hamster, one's from a mouse. Okay, hamster, mice, they look alike. And all of a sudden, there was a big explosion in research. Everybody started doing these experiments, everybody. And then Weinberg's lab came out with another paper very soon thereafter, the next year in 1982, Nature, volume 297, page something. I can't see the page. Well, okay, 474. So what did they find? They found that at least one of the genes being transferred from the cancer cells to the normal cells was equivalent to a viral gene called Harvey-Raz. Now we know it as HRAS. So there's a Harvey-Raz virus, and the cellular gene homologous to the Raz gene of the virus was thought to be at least one of the causative agents when DNA was transferred from malignant cells to non-malignant cells. So all of a sudden, we didn't need viruses anymore. The cells had their own oncogenes. We didn't need the virus to cause the cancer. Somebody just asked for a list of the papers. I'd be happy to provide it. Best thing to do, you can email me. My best email address is all@anticancer.com, all@anticancer.com. Anybody can email me. I'm happy to hear from everybody. I'm happy to give the list or anything else, happy to talk. It's my pleasure. So then another story started to happen. There are a couple of papers, also in Nature, followed very shortly. At the end of 1982, Nature volume 300, page 143, that's from Weinberg's lab, and Nature 300, page 149, I think one paper following another, from a laboratory of Dr. Barbacid, and they found that the transferred RAS gene had a point mutation in it. So people said, okay, the cancer cells become malignant because the oncogene that resides in those cells, or gene then, got a mutation and then became an oncogene. So the cells have genes that are homologous to what are found in viruses. Some of these viruses can cause malignancy, at least in cell culture and maybe some animals, but never been found to cause cancer in a human yet. And the theory went, okay, if these genes get a point mutation, then they become a true oncogene, and that's the basis of cancer. And so all this was attributed to Dr. Weinberg. Spondinos got airbrushed out of history, like they do in the Soviet Union. They take a picture and they airbrush the picture, and the person in the picture disappears. So that's called airbrushing. Spondinos got airbrushed, really got airbrushed. Some people say it's not. Okay, there's some notes coming up. I don't know. I hope everybody can hear. And if you can't hear, I'm sorry. But anyway, that's where we were. So Dr. Weinberg was the leader of this, and his theory was you could get cancer from a single mutation in an oncogene. It was pretty amazing because the mutation rate is about 10 to the 6th, minus 6, and even a baby newborn must have 10 to the 12 cells. So how could a newborn be born without cancer? But nobody seemed to care about that. It was one gene, a mutation in one gene, the RAS gene. That was the cause of cancer in 1981, like that. And so this theory never died. So a few years later, they said, okay, the dominant paradigm went from one gene can cause cancer to 2 to 3. It started increasing. And then Weinberg and some other people discovered what they called tumor suppressor genes. So you had oncogenes and tumor suppressor genes, and if the suppressor got mutated, then the cell would get cancer, maybe whether or not the oncogene got activated or not. This was the theory of cancer in the 80s, in the 90s, even into this century, and it started to become known that the cancers had thousands of mutations. Which ones were the cause? Which ones were the cause? So this could not be explained. So the paradigm switched a little bit from oncogenes to driver genes. that's where we pretty much are now, although Warburg has become back into fashion again, and cancer metabolism has become an area of interest again. Of course, glucose is the main interest in cancer metabolism and how it's altered. And on the very periphery is methionine metabolism. So I have to tell you how I felt back in 1976 when we published this paper. I knew that this was important, and I knew that this methionine addiction was important for cancer. I knew it way back then, but this was a time of a period of time when the viral carcinogenesis program was maybe at its peak, and it was just the beginning of the oncogene era. And I felt very overwhelmed by those paradigms and all the powerful people that followed those paradigms. So it was very hard. I thought I was very peripheral, my work, and it was very, oh, somebody wants my email again. Okay, I'm sorry. Thank you for your note. It's all, ALL, at anticancer, ANTICANCER.com, all at anticancer.com. And I'll answer everybody. It would be my honor and my pleasure. Please email me. I've got lots of stories to tell, even more than today. Today's an overview, an introduction. So I felt overwhelmed, and I even changed some of the titles of our papers. So, I didn't want to mention methionine metabolism. I thought, well, we'll just try to be more in the general view, even though the papers were on methionine metabolism. So let me tell you what happened subsequent to our first paper in 1976. And I want to give much, much, much credit to my mentor at that time, Richard W. Irby, ERBE. He was in the lab of genetics that was called at Massachusetts General Hospital. That's where I did my postdoc. I was there for 6.5 years. It was a wonderful time. And Dr. Irby, with great patience and with great knowledge, introduced me to methionine metabolism. And I thought, oh, wow, this is so fascinating. And I saw a paper in PNAS. It said, oh, cancer cells need methionine because they can't make it. And I don't know why, but I didn't believe it. I didn't believe they couldn't make it. And we did experiments on the cancer cells, 3, 4, 5. Yes, you ask if you can change the slide. Sure, change the slide anytime. You want to change the slide now, Ashley? You can change it. Okay, yeah, that's right.
Hello? Are you there, Dr. Hoffman? Hello?
Offline. Hello?
Can you hear me, Dr. Hoffman?
Yeah, I can hear you. Can you hear me?
Yeah, I can hear you.
Oh, we're back. That's good. I didn't want everybody to go away.
No. I think there was a slight glitch in the network.
Little bird. I heard it go tweet, tweet, tweet. Anyway,
all right. We can continue.
Okay. So we studied the biosynthesis of methionine in a number of different cancer cells. Methionine synthesized from its immediate precursor called homocysteine. It just needed to be methylated homocysteine. And we found very high activity in the cancer cells, yet they would not grow without exogenous methionine, a condition in which normal cells grew very well. Normal cells didn't need exogenous methionine. They loved homocysteine. As long as there was some B12 there, some folic acid, they did extremely well. But the cancer cells, they couldn't do it. They couldn't grow without preformed exogenous methionine, even though they were making tons of methionine from homocysteine, as much as the normal cells, maybe more. And previous to that, as you can see from this slide, Dr. Sugimura, who became the most famous cancer researcher in all of Japan, he just passed away this year. He was about 94. And in 1959, when he was visiting at NIH, he studied rats, and they had tumors, and he took away one amino acid at a time from their diet and found that the amino acid that made the difference was methionine. Well, that nobody cared about that. That stayed in some buried under whatever, even though it was published. And some years later, Dr. Cello and Dr. Bertino found that leukemia cells did not grow when homocysteine replaced methionine. So finally, we could study this in cell culture because of Cello and Bertino. And Dr. Bertino is still a great oncologist. He's 90 years old. He's active, doing all kinds of research and very interested in methionine metabolism, and has encouraged me for 40 years. Okay, Ashley, next slide, please. Okay, well, okay, all right. So this addiction for methionine that cancers have, addiction for exogenous methionine, well, it's called the Hoffman effect. That's nice. But whatever you call it, it is what it is. And it was a mystery why the cancer cells needed so much methionine, and basically it had to be exogenous methionine. Well, back then there was a man in the lab, in our lab, when I moved to UCSD in 1979. I've been at UCSD for 41 years. That's a long time. And a man in the lab called Peter Stern. Now, I knew Peter a long time, since I was an undergraduate at the University of Buffalo. One day I just saw him in the street in San Diego. Years and years and years later, 15 years later, I said, "Hey, Peter, what are you doing here?" He didn't recognize me, of course. And one thing led to another, and he joined the lab. And he made a very fundamental discovery. It's here, and I'll read the title: Elevated Overall Rates of Transmethylation in Cell Lines from Diverse Human Tumors. Now, that's a lot of words in just 30 seconds, but what he found, very interesting, what Peter found way back then. This is published in the journal called In Vitro, volume 20, 1984, page 663, and this particular paper was edited for the journal by another one of my great teachers, Gordon Sato, who passed away a couple years ago. A great teacher, and was so kind to me. And what Peter found in this paper, I'll show you the graph. So here's a graph, and this graph, all the high peaks are transmethylation rates of different cancer cell lines. The normal cells are the dashed lines way at the bottom. So every cancer cell that we looked at had a high rate of transmethylation compared to the normal cells. This is a transmethylation rate there. So we got a hint. That methionine addiction was due to the overuse of methionine for transmethylation reactions in the cell. This paper was published in 1984. And for 35 years, we didn't know where all these methyl groups were going, why the cancer cell has all these excess transmethylation reactions. That's what was using up all the methionine. And you see in this very cute cartoon, which comes from A-Star Research, which is, I think, one of the funding agencies for research in Singapore. And you see the poor little guy that's using glucose. Well, he's a small little guy. And then you see this great big guy using methionine. And so that's the cancer cell. The cancer cell is more hungry for methionine than it is for glucose. And how do we know that? We know it from the clinic. So I mentioned a little bit that radioactive analogs of glucose are used for PET imaging, positron, electron, something, PET, that's good enough. And it's a very sensitive way to light up cancers because they take up so much glucose and it's radioactive and you can see, and very good diagnostic for the patient. But a number of clinics all over the world are now using carbon-11 methionine for PET imaging. And when we call PET-MET, PET imaging with methionine is compared head to head in the same tumor, maybe same patient, with glucose PET. Methionine is always the winner. You get a stronger and more specific signal in the cancers with radioactive carbon-11 methionine. Why? Because the cancer is so hungry for methionine, it's an addict, like a cocaine addict. He's got to have a lot of cocaine. The cancer cell has to have a lot of methionine. And we had the basic answer, but not the specific answer, that this addiction was due to excess transmethylation reactions in the cancer. But for 35 years, we're wandering in the desert like Moses coming out of Egypt. We didn't know where these methyl groups were going until about 1.5 years ago. And a paper was published in Singapore in Nature Medicine. First author is Wang, W-A-N-G, and the senior author is a fellow called Tam, T-A-M, Wei Leong Tam. I'm sure I'm mispronouncing that, and forgive me. That's Nature Medicine, volume 25, 2009, page 825. And the title of this paper is Methionine is a Metabolic Dependency of Tumor-Initiating Cells. Okay, and in this paper, they compared what they called tumor-initiating cells, and these were cells in culture that grew in suspension, and they compared them to what were called cancer cells that were not tumor-initiating, that grew attached in the plate. And this paper showed that the flux of methionine was very elevated in the tumor-initiating cells, just like we showed in the cancer cells and the cell lines we studied 43 years before. And then this paper said the excess flux of methionine in the tumor-initiating cells was due to transmethylation reactions. And they also showed there was a difference in the methylation of histone H3 between the tumor-initiating cells, which had the high methionine flux, and the non-tumor initiating cancer cells. Here's a little figure here. And you see this is Western blots. These are all different methylation marks. Here, I'm sorry if it's hard to see. But anyway, they compared the methylation of histone H3, different lysines, and they had much more methylation in the tumor-initiating cells than in the non-tumor initiating cells. If you can see the black marks by my finger, those are the tumor-initiating cells, and right here are the adherent cells. So black marks mean lots of methylation, and so we got a good hint. We got a hint we were waiting for 43 years. Maybe that was the difference between not only tumor-initiating cancer cells and other cancer cells, maybe that was the difference between cancer and normal. So we read this paper and we got really excited. I wrote a letter to Dr. Tom. I congratulated him and everything like that. And so this was a hint. Maybe we're going to find our way out of the desert into the promised land of methylation. Okay, so we started to study histone methylation in cancer cells and in normal cells. And but I want to mention one thing before. About in 1980, we were studying the methionine dependency of cancer cells and where they arrested in the cell cycle when they were restricted of methionine. And we found that they arrested in late S and G2, which is very unusual. And then we found another thing, and we didn't realize the importance, that when cancer cells were restricted of methionine, this is back in 1980 now, it was published in PNAS, the cells, the cancer cells, lost their ability to clone. You put them back in methionine, no clones. If they were always in methionine, lots of clones. So this was very, we thought, oh yeah, okay, we thought that was interesting. But now going back, we realize maybe those clonogenic cells were equivalent to the tumor initiating cells. They die upon methionine restriction. We thought back again, getting the hints from this paper. So we started to study then histone H3 methylation in cancer cells, normal cells, and another kind of cell that we worked on, that we call them revertants. And these are rare cells that you could select from the methionine addicted cells that became methionine independent. And we first studied this back in the late 70s. It's titled Reversion to Methionine Independence in Simian Virus 40 Transformed Human and Malignant Rat Fibroblasts is Associated with Altered Ploidy and Altered Properties of Transformation. So that's a lot of blah, blah, blah information. This was in PNAS, volume 76, page 1313, 1979. So what we, and I'm happy to send all these lists to you, my pleasure. Oh, here's a message from Russia. Oh my gosh. I could read a little bit Russian, but the message was only a little bit, was quick, and I'll just say to that nice Russian person who sent the message, I'll just say privyet to you and thank you. So these revertant cells were no longer methionine addicted. They could grow on homocysteine, and years later we found that if we restricted methionine with an enzyme we worked on and purified and trying to develop it into a cancer drug called methionase, we could restrict methionine with methionase, and the revertant cells were quite resistant, much more resistant than their methionine addicted parents. So, in addition, back then, we weren't doing any tumor studies in the mice. But we found many aspects of malignancy that were associated to malignancy in vitro became more normal. So if you select cancer cells to become non-methionine addicted, to become methionine independent, they can grow on homocysteine or in very low levels of methionine, they started to become normal. So we knew methionine addiction and cancer was linked. And so that's where we are now. And I want to tell you the current story. And I want to bring my young student who's leading this research. If you just bear with me 1 second, let me call him. June, where's June? Ask him to come here, please. Okay, so I've just asked Jun Yamamoto to come here. And I'd like to introduce him to all of you because he's doing all these experiments comparing histone methylation in cancer and revertants and in normals. And it turns out that the revertant cells and normal cells do not have hypermethylation. And when it goes away, when cells revert toward normal, they lose their tumorigenicity. So there's, where's June?
Yes.
There's June. Okay, June, get a seat, please. Okay, hang on. I want to introduce you to June. He's a young man. He's less than half my age. Here he is. This is June. June is from Yokohama City Medical University in Japan. He's been working in our lab for about 2 years. He's made a great breakthrough. And this is June's new paper here. It just came out on the preprint server. It's very, very hot, only 8 days old. It's called Cancer-Specific Overmethylation of Histone H3 Lysines is Necessary for Methionine Addiction and Malignancy. Okay, that's a lot of words here, but it means that our results suggest that if the cancer cell doesn't have overmethylated histone H3, it's not malignant. So does that mean overmethylation of histone H3 is the cause of cancer? Maybe. And histone H3, there's many marks of histone H3 that are overmethylated in cancer. And these marks can serve as both inducers and repressors of gene expression. And we're asking, is this the basis for cancer? So June, you want give a little bit summary of your work, please? A couple sentences to the people.
Okay. Hi, nice to meet you. I'm Jun Yamamoto.
Why don't you tell them the basic summary of what you found?
Yes, so we found the difference of the histone methylation status between normal cells and cancer cells. And the cancer cells has overmethylated histone H3, but not have, but normal cells doesn't have overmethylated, low methylated histone H3. So it means maybe the gene expression is totally different between normal cells and cancer cells. It may be. And we also found we established the methionine independent revalent cancer cells from methionine addicted cancer cells. So the origin is same cancer cell lines, but the methionine independent revalent cancer cells has lower methionine levels compared to parental cancer cells. So that means methionine independent cancer cells lost the tumorigenicity and its malignancy. We think. It's
okay, very okay. Thank you, June. And I'm afraid it's a little bit late. It's question time now. So I can stay as long as Inago lets me. I'm happy to try it. June and I will be happy to answer questions. So, okay, Ashley?
Can you hear me, Dr. Hoffman?
Yeah, we're ready for questions now.
All right. Just give me a minute.
I'm sorry, we went a little bit too long, but I can stay later. I can stay.
All right. Just give me a minute, Dr. Hoffman.
Okay. Some people want the slides to be emailed. We can do that. We can do everything.
All right. So there is a question at the moment, Dr. Hoffman, from one of the attendees.
Okay. Can I have the name, please?
Yeah, the name is Pranati Gandhi. That is the name of the attendee. Pranati Gandhi.
Gandhi? Dr. Gandhi?
Yeah. Gandhi,
like Mohandas? Mohandas Gandhi. Okay.
All right. The question is, we know that methylation is an epigenetic mechanism. So is this, does this have an effect with DNA sequence? Why is cancer inherited among generations?
Why is cancer what? I'm sorry?
Why is cancer inherited among generations?
Okay. So some cancers are inherited, mostly are spontaneous. Of course, there's some propensity to cancer in families, but inherited cancer seems to be due to some inherited genes, but this is not typical of the vast majority of cancers. So the inherited cancers are a special situation, and they can be studied by studying specific genes. We don't think spontaneous cancer can be studied by just specific genes only, like the paradigm for 40 years has suggested. We think there's massive changes in gene expression and even in mutation in cancer, perhaps due to the fact that histone H3 methylation is so altered. That doesn't mean that's the only reason for cancer, but we think it's an important reason. So I'm sorry I didn't give a very great answer, but I did the best I could. I'm ready for the next question, or June is ready.
All right. All right. The next question, Dr. Hoffman, is which enzyme is responsible for the transmethylation of histone 3? Only H3? How about H2A, H2B, and H4?
All right, I'm going to just discuss with June 1 second. June, I think there's many transmethylases involved, right?
Yeah.
And they're specific for each mark, right? Like HK4.
Yes, each mark and each methylation level. So from monomethyl to dimethyl or dimethyl to trimethyl. So there are so many methyltransferase or demethyltransferase.
Also, so it's very complicated, and so many transmethylations. Maybe Anna Puma wants to send me a message. So many transmethylases are involved in this, and maybe that's part of the reprogramming of methionine metabolism that is the basis for this excess methylation. It may be a so-called reprogramming effect where many different transmethylases, and not only transmethylases, get activated or inhibited. So I think it's a fruitful area to study, but one, like June said, as June said, maybe many transmethylases are involved. Next question, please.
All right, just give me a minute, Dr. Hoffman.
Sure, take your time, Ashley. They want a few more slides. You can show the slides. Anything you want, you can do it. Okay, Ashley, maybe, okay, when we end, at the end of questions,
Yes.
we go through the slides one by one. And
Sure.
if with permission of Vinago, we can email the slides to people who want them,
Sure.
and we're happy to do that. There's a question from Malaysia. Oh boy, good.
Yeah,
Okay.
so there's one more question. Today's cancer is the second most disease-causing agent of the deaths in the world. Was the statistics always like this, or is there another reason for it?
I'm sorry. Is why is cancer so frequent?
Yes, I mean currently cancer is the second most number of deaths. It causes the second most number of deaths in the world. Why was this statistics always like this back in the days, or is there another reason for it?
Okay, so this is an important question. So 100 years ago, most people didn't live long enough to get cancer. They died of infectious disease. So cancer was rare. And also 100 years ago or 120 years ago, people didn't smoke. And when lung cancer was very rare, and when people started to smoke and the lung cancer rate started going up, the doctors would bring their students to see a lung cancer patient because they never saw one before. So smoking made the cancer rate go way up. And aging, so people got older. They didn't desire. Antibiotics were developed, so people did. In 1900, in America anyway, the life expectancy was 48 years. Now it's 80. So people lived long, And the longer you live, the greater the chance that you get cancer, because probably many things have to happen inside the cell in order for it to become a real cancer. Dr. Duisberg says a cancer is like a different species even. It just gets evolved in the body. So cancer takes time. If you die young, you're not going to get it. You live long, there's a greater chance to get it. People say almost every man gets prostate cancer, whether he knows it or not, if he lives long enough. So, and also some of our lifestyles not good. Smoking is really bad for cancer. Now, at least in America, so many people are obese. That's a big risk factor for cancer. So the cancer rate goes up. Of course, we're better at diagnosing. We're a little better at cure, not too much better. We can catch it more early, and that's good. So I think that's at least part of the answer to our colleague from Malaysia.
All right. There's one more question. May I know the ideal level of methionine required by the body?
Okay, this is a very tricky area. So generally, the methionine level in the body is about 30 micromolar. Probably this is a big excess. So in the, oh, Steve, thank me. Thank you too, Steve. Thank you for coming. Steve Clark, great scientist, great friend, classmate. So in rodent studies, not by us, there's this fellow called his name was Orentreich, and he invented hair transplants, and he became a zillionaire, and he used his money to study methionine restriction in rodents, and he found that he could increase the lifespan of a rodent 30% by restricting methionine. So the methionine level would go down. So 30 micromolar in a human body is probably too much. And if you restrict methionine in rodents, you can reverse obesity, prevent obesity. You can prevent and reverse diabetes, maybe even fatty liver, maybe a whole bunch of other things, maybe autoimmune disease, because T cells require lots of methionine to be activated to cause multiple sclerosis and things like that. So what does the body require? How much does the body require with regard to methionine? It depends. And it looks like low methionine is good. If you can lower your methionine, people who are vegan eaters, that's pretty tough to be a vegan eater, have low methionine, and maybe they're going to be more healthy. Maybe they're going to live longer. Maybe they're not going to get cancer. Or you can take, now our methionase can be taken orally. We have given it to some people as a supplement, not a drug, and they lower their methionine level. So probably 30 micromolar is too high. Maybe 10 micromolar is too low. We don't know yet. We did studies not on oral methionase, but injectable methionase 20 years ago. And we gave the macaque monkeys injectable methionase, and they got reversible. Their hemoglobin level, they became anemic. So what's the optimal methionine concentration in the body? It's probably not the 30 micromolar we find in general. Probably lower methionine is good. Lowering methionine may, and it seems to, mimic calorie restriction in animals anyway. And that has so many healthy effects. The methionine restriction as well as calorie restriction make the biomarkers of metabolic syndrome go away. So maybe you don't have to starve yourself all the time, just swallow a little methionase once in a while. So this is a very important question, and we got a lot to learn. All
right. Dr. Hoffman will take one last question, and then by special demand.
Don't disappoint the people, Ashley. Don't disappoint them.
No. In fact, actually, by special demand, the audience want to go through the slides again. So we'll take one last question, and then
we do anything for the audience, Ashley. Without the audience, we wouldn't be here.
All right. So there's this question: iPSC is also methionine addictive and hypermethylated. Do you think it is related to your hypothesis?
I'm sorry, what's methionine addicted?
IP, IPSC,
I,
IPSC, IPSC.
oh, iPS cells,
Yeah, IPS is also
induced
methionine.
pluripotent stem cells, IPS.
Yes.
Okay, I don't know about that, but I suspect the level of methionine can greatly influence the differentiation potential of those kind of stem cells. And I'm sorry to give a very uninformed answer, but I can recommend looking this up, checking it out, and also doing some experiments with IPS cells under methionine restriction. It might be extremely interesting. I think some people have done it a little bit, just beginning, and I don't remember much at all. But I think it's a very fruitful thing to study, the methionine requirement of IPS and other kind of stem cells. I think it's very exciting. I think
All right.
methionine is the most important molecule in the whole body.
All right. Yeah. So, as requested by our audience, Dr. Hoffman, we'll just quickly go through the slides,
Okay,
before
please
we
go
wrap up.
ahead, Ashley.
Yeah, just give me a minute.
I'll take your time.
I think we can continue from the slide that we left off in on the Hoffman effect.
Yeah.
This was the last slide. I think
I'm sorry, I got too excited to say,
That's
tell you
all-
the slides. I'm sorry.
that's all right. I think we can go on from this slide quickly through the rest of the presentation.
I think we have to give the audience the slides, Ashley. They
Yes,
deserve it.
we will be sharing the slides as well as a recording for this session.
Good. God, please, the audience has been so wonderful.
Yeah, after 1 week, it'll be available on our website.
Great.
And any further questions that are given by our participants, we will share with you, Dr. Hoffman, over email.
Okay, and people can
and-
email me directly too, all at
Yes.
anticancer.com. My pleasure.
Yes, and we can get that addressed to you as well.
So I would like to end. Jose Hernandez says, thanks. You're welcome. So I would like to end this. No, I never want to end, but we'll call this a comma, not a period.
All right.
That science takes time. Methionine addiction is still not, I would say, maybe far from the center of current cancer research, but it's making inroads. It's getting there. And it's also the methionine addiction of cells is a great therapeutic target. Restrict methionine. Now, it's getting popular to restrict methionine by diet, but that's tough. You got to be a vegan, and it's not easy to be a vegan. But maybe methionase is a less onerous way. We can target methionine addiction. We've been trying to do this for 25, 30 years. And of course, it's not central to cancer therapy either. Now everything's immuno-oncology. 40 years ago it was immuno-oncology. It's cycled back. So metabolism is getting more interesting. 100 years after Warburg's 1924 paper. So we'll see what happens. It takes time. It takes time. And in my opinion, cancer research is not yet a solid science. It's getting there. I don't know how much longer it's going to take. Astronomy became a science in the 1500s after Copernicus discovered the planets go around the sun. Before that, it was not astronomy, it was astrology. So I'm hoping that cancer research will become a more solid science, not just based on what's popular or what's based on what the big boss does, what's fashionable. If we can just go step by step and keep the science solid and always rooted to the clinic, or what we see, to what we see in vitro, to what we see in the animals. Does it reflect what happens in the clinic? This is really important. The clinic is our denominator. We have to always check ourselves to see if what we're studying has relevance to clinical cancer. And when we start doing this on a regular systematic basis, cancer research will become a really solid science. I think it's going to happen. So I think I've been told it's enough now.
All right.
It's
No,
never enough for me. I'm sad to
true,
stop talking to you.
true,
I'm so
true.
happy all these wonderful people came to the talk. Send me lots of emails. I'm your
Sure.
friend forever.
Sure. Thank you, Dr. Hoffman, for this very
Thank you,
insight.
Ashley. Thank you,
Very,
Inago. And biggest thanks to the audience. Thank you, thank you, thank you.
thank you for this very insightful and informative session. I'm
You're
pretty sure.
all welcome. Stay in touch with me. I'm your friend
Sure.
forever. Please.
Sure,
Thank you.
definitely. I'm
Thank
sure
you
our
all.
I'm sure our
And
attendees
I want all of you to make a great contribution to cancer research. We need it.
after this session, I'm pretty sure they will. Those who are working in the field.
Thank you, Ashley Fernandez. You have a
Thank you.
Portuguese name.
Thank you so much. All right.
Okay.
I'm sure our attendees have gained a lot of crucial information from this session. I'd like to
Okay.
thank you a lot for taking the time out from your busy schedule to share this information with us and share your insights on cancer research and methylation in cancer. All right. For all attendees, I'll request you to please fill up the feedback survey displayed after you leave the session. Your participation will allow us to evaluate the effectiveness of future webinars. And, I hope you all had a wonderful session. Thank you, Dr. Hoffman, once again for your
Thank
time.
you, Ashley. And thank you, everybody at Inago, to make it happen. Thank you to Ms.
Thank you.
Hozumi, who helped an awful lot too.
Thank you.
Couldn't do it without all of you. All I know how is to talk. I don't know how to use a computer. I just know how to talk. So I really appreciate you made beautiful slides. You did everything right. You got a whole lot of audience around the world. It's wonderful. Thank you all.
Thank you very much, Dr. Hoffman. Have a wonderful evening. Have a wonderful evening to my audiences as well. Thank you.
Nap. You had to get up very early today. Have a nice nap after lunch.
Yeah.
We'll talk to you soon. We'll get
Sure,
I'll respond to everybody's email, and we
Sure.
want to send everybody the slides, everything. We want to be really everything the audience wants, we have to do.
Sure, definitely. So just a small announcement to our audiences. Any questions that you have shared with us, we will get in touch with Dr. Hoffman and share the answers with you over email. As well, the recording for this webinar and the slide will be available on our website, Inago Academy, within 1 week. So you can definitely come back and watch this recording all over again at your own convenience. Thank you again, Dr. Hoffman. Thank you once again to all our audiences.
Thank you, Ashley. You're welcome.
Have a great...
It was my pleasure, my honor.
Same here.
Thank you.
Same here as well.
I'm glad you can meet June, because June's going to take over from me. He's the future.
That's great. Thank you very much. Have a great evening, Dr. Hoffman.
You have a nice day, a nice Wednesday. We're still on Tuesday.
Thank you. Yep.
Bye, everybody.